Hello everyone, welcome back to our videos on Form 5 Biology. In this video, we'll continue to discuss the topic from Chapter 1, that is 1.5, Role of the Circulatory System in the Body's Defense Mechanism. We are going to talk about the types of immunity. In the last video, we discussed active immunity, so in this video, we'll end by discussing passive immunity. The learning outcomes for this video are as follows. First, we must be able to state the definition of passive immunity. Secondly, explain how the following types of immunity are acquired and make a comparison. Passive immunity, and then a comparison between natural passive immunity and artificial passive immunity. Number 3, state the examples of diseases related to each type of immunity. And number 4, explain the graphs related to the two types of immunity, which is natural passive immunity and artificial passive immunity. Let's revise this again. What is immunity? Immunity is the ability of an organism, of a human being, for example, to resist infection or disease. And that's by defending itself against infection by pathogens. For example, when pathogens enter the body, what do our lymphocytes do? And whether they can overcome the infection. So immunity depends on the presence of lymphocytes, the special white blood cells, which produce specific antibodies during infection. And this is called a specific immune response to infection. So a person with a strong immune system is one who is able to resist the infection and not fall sick. But he can overcome the infection and destroy the pathogens that have entered his body. Let's look at the overview once again. I would suggest that you can copy this in your notebook to help you understand the overall picture, because this picture helps you to connect all the concepts together. So immunity is divided into two main types, active immunity and passive immunity. We've already studied active immunity, but we just revise again. It's when a person's own body produces his own antibodies when stimulated by antigens, either through injection or the antigens may enter the body naturally and cause a person to fall sick. So a person actively produces his own antibodies. His lymphocytes will produce the antibodies. But in passive immunity, the body is passive in that it does not produce the antibodies. It just receives the antibodies from an external source, like from the mother or from injection. So active immunity can be divided into natural active immunity and artificial active immunity. For natural active immunity, it's obtained when a person falls sick after being infected, like fall sick from chickenpox or measles or even influenza. Then he starts to produce his own antibodies because the lymphocytes will be stimulated to produce antibodies. And finally, when the antibodies reach a high level, beyond the level of immunity, he recovers. After that, he has memory cells, which are lymphocytes, that can produce the same specific antibody again, should there be a second infection. And when the second infection occurs, the production of the antibodies are very fast and also in great amounts. And this will destroy the pathogens so that the person doesn't fall sick at all. So he's said to have natural active immunity against the disease. For artificial active immunity, the word artificial means there is an injection involved. So vaccine, which are dead antigens, dead or weakened antigens, is injected into the body to stimulate the body to produce its own antibodies. So the person will not fall sick because this vaccine contains dead or weakened antigens that don't harm the body. But the aim of this vaccine injection or vaccination or immunization is to help the lymphocytes, to stimulate the lymphocytes to produce that specific antibody against the antigens, so that in future, should the living antigens attack the body, then the memory cells produced will be able to produce the antibodies specific antibodies against that antigen and overcome the attack, and the person will not fall sick. So that's artificial active immunity. In both these cases, memory cells are produced that can later produce a more rapid response, immune response, should the antigens enter the body the second time. So the person will not fall sick. Now, today we'll study passive immunity, which is divided into natural passive immunity and artificial passive immunity. Now, for natural passive immunity, a baby or fetus will naturally receive antibodies from the mother or the pregnant lady. So through mother's milk, or for pregnant women, they will give the antibodies through the placenta. We'll discuss more of this later. And for artificial passive immunity, once again, the word artificial means there's an injection, and this time it's an antiserum containing the ready-made antibodies that are injected into the body of a person who may be sick already and may have pathogens in his body. So these antibodies are injected to destroy the pathogens that have entered the body. Let's discuss the two types of passive immunity. The first type is called natural passive immunity. It is acquired when the mother gives the antibodies to the fetus in the uterus through the placenta and to her baby after birth through the breast milk. So here we have a picture of a pregnant woman with a uterus, and inside we can see the placenta, which is an organ that consists of the maternal tissues and the fetal tissues. So the placenta provides the fetus with oxygen, nutrients, and also antibodies. The antibodies will diffuse from the mother's blood into the fetal blood in the placenta, and then the antibodies will be transported to the fetus. And it will protect the fetus in the mother's uterus too for some time, until the birth of the child. So when a baby is born, the mother can continue to provide that kind of protection, natural passive immunity, for the child through the breast milk. Especially in the first few days after birth, colostrum, which is the first mother's milk produced in the first 3 days, is provided to the baby and is rich in antibodies to help protect the baby who is newly born and may face attack from pathogens in the environment. So this colostrum is important for the baby to have, and that's why mothers are encouraged to breastfeed their children right after they are born. Secondly, the maternal antibodies give only temporary immunity to the baby for 6 months. The reason is to protect the baby from disease until the baby's immune system is fully functional. So when it's first born, the baby's immune system is not well developed yet, and it has to become fully functional. So between that time is the maternal or the mother's antibodies that help to provide temporary protection to the baby from certain diseases. However, this immunity is only temporary. Why is it only temporary? Because the mother's antibodies are foreign to the baby. So they are broken down in the baby's body and they are not replaced. The baby's own lymphocytes do not provide the antibodies. So therefore, after the antibodies have been broken down, they are not replaced. And the baby will then have to develop its own immunity against various diseases. Let's discuss the graph for natural passive immunity. This graph shows you the concentration of antibodies against time. And the level of immunity here is the concentration of antibodies that must be reached by the baby in order to become immune towards a particular disease. Let's take the case of a mother breastfeeding her baby. So as the breast milk or mother's milk is provided, in it there will be the antibodies too. So the level of antibodies that's provided to the baby will increase And the concentration of antibodies will increase very fast because these are ready-made antibodies from the mother supplied through the breast milk. So as the concentration of antibodies exceed the level of immunity, the baby will become immune to the particular disease. However, the concentration of antibodies also decreases very rapidly because the antibodies are foreign to the baby as they are from the mother. Thus, the antibodies will be broken down in the baby's body and they are not replaced because his lymphocytes did not produce these antibodies. They came from an external source. So the graph is like a steep curve that goes up and then comes down very fast. And after about 6 months, the level of antibodies from the mother will drop to 0 when the baby's immune system is more fully developed. Let's look at another graph. This graph shows you the graph for natural active immunity, not passive. So if you compare the two graphs, let's take the case of the baby falling sick from some flu or cough. So when he falls sick, firstly the pathogens will enter his body and this will stimulate his own lymphocytes to produce the antibodies, which will increase very gradually as it is the first infection. And after it exceeds the level of immunity, after the concentration of antibodies exceeds the level of immunity, the baby becomes immune to the flu or the cough. And then his level of antibodies will also decrease very slowly or gradually as his own lymphocytes are the ones that produce the antibodies. And he also has memory cells that can help to produce a more rapid response in case there's a second infection. Okay, so the graph is gentler, the slope is gentler compared to the graph for natural passive immunity. Let's discuss the second type of passive immunity, which is artificially acquired passive immunity or artificial passive immunity. When we have this word artificially acquired, it means that it involves an injection of a substance. And in this case, it is injection of antiserum. So this type of immunity is obtained or acquired when a sick person is injected with an antiserum or a serum containing antibodies prepared from the blood of animals. For example, a person may have been bitten by a dog that is suspected of having the disease called rabies. So he goes and sees a doctor, and the doctor will give him an injection of the rabies antiserum, which contains the antibodies for rabies to destroy the virus that causes rabies, which is suspected of having entered his body. So the doctor will inject the antibodies at a certain concentration which is above the level of immunity, so that the person will be able to become immune and destroy all the pathogens that cause this rabies. So this type of injection is given to a sick person or a person who is suspected of having been infected with the pathogen. And the name of the substance that's injected is called antiserum, meaning serum containing antibodies. So the word antiserum actually means antibodies in the serum. Okay, and serum means blood plasma without fibrinogen. So how do we get the serum containing the antibodies? It must be prepared by using animals which are intentionally injected with these antigens or pathogens or the toxins so that they fall sick and start to produce antibodies for that specific disease. So after they recover, their blood will be extracted and it will contain the antibodies which can help sick people recover. So this serum containing antibodies will be sent to the hospitals and clinics. In case there's somebody who is sick with that particular disease, they can be given the antibodies straight away. through injection to help them recover. Okay, so this antiserum destroys pathogens that have already infected people. It's unlike vaccines which are injected into people who are healthy. Number 3, it gives quick but temporary immunity. So quick immunity because the antibodies are injected into the blood at a high concentration above the level of immunity to give immediate immunity to the person. But it's temporary because these antibodies are from external source, like from the blood of animals. So they are foreign antibodies. And since they are from the external sources, they will break down in the body very quickly and they are not replaced. Because the lymphocytes of the person did not produce the antibodies. It was from an external source. Here are some types or examples of antiserum that have been developed for various fatal diseases. So all this antiserum contains the antibodies for that particular disease. Rabies, or sometimes called mad dog disease, is a viral disease transmitted through the bites of wild animals, and this virus attacks the nervous system. Botulism is caused by a toxin from a bacteria that also attacks nerves, and it is transmitted through food that is not well preserved, like canned food that is not well preserved may contain this bacteria which produces the dangerous toxin. Tetanus or lockjaw is caused by bacteria in soil, a certain bacteria that produces a dangerous toxin too, and normally is transmitted through the cuts and wounds caused by rusty objects like nails and other objects in the soil, for example. And the last one is snake bites that contain toxins from the particular snake. Let's have a look at this graph for artificial passive immunity obtained through injection of antiserum. So the graph is very different from the one for injection of vaccine, for artificial active immunity. This graph goes straight up because when the doctor injects the sick person with the injection of antiserum, in the first injection, he will make sure that the antibody level is very high and beyond the level of immunity in order to destroy the pathogens effectively, the pathogens in the body. So after that, the level of antibody will decrease very fast because these are foreign antibodies that are broken down by the person's body. Next, the doctor may give a second injection to boost up the level of antibodies beyond the level of immunity again. So it's called the booster dose of antiserum. And this is again to destroy any remaining pathogens that may be able to still cause harm to the patient. Okay, so the person will have temporary immunity from the disease. And the level of antibodies decreases very fast compared to artificial active immunity. So the graph is something like shark's fin under water like that. So remember the shape. This is for artificial passive immunity. The shark's fin shape, right? For artificial active immunity, the graph is like two peaks, one lower and one higher one. Okay. A common question asked in the exams is, for artificially acquired passive immunity, why is a second dose or booster dose of antiserum needed? So if you look at the graph here for injection of antiserum, this graph on the right, you can see that after the first injection is given, the level of antibodies decrease very fast below the level of immunity. So a second injection is given at this point. So why is it given? So it is needed to increase the concentration of antibodies so that it exceeds the level of immunity again, as we can see here. This is to destroy all remaining pathogens even as the person acquires the artificial passive immunity again, right? So the main reason for booster doses are to make sure that all the remaining pathogens are destroyed at the high level of or high concentration of antibodies. Now this is unlike artificially acquired active immunity, as can be seen in this graph here. See, the booster dose is given somewhere here, is to raise or increase the level of antibodies or concentration of antibodies to exceed the level of immunity for the first time, and that is to help the person acquire the artificially acquired active immunity. It's not to destroy pathogens, because this person did not have pathogens in his body that caused him to be sick. Okay, so we must remember that the reason for the second injection is different for injection of vaccine here and for injection of antiserum here. Okay, that's all for this video. Do try the question that I've uploaded. It's a video on how to answer essay questions, and the question is based on types of immunity. Do try that question because it is a popular question for the exam and it has not been asked so often these past few years. In the next lesson, we'll continue to discuss Chapter 1, Part 2 about transport in plants, especially about the xylem and phloem tissue and other topics related to it like transpiration and opening and closing of the stomata. Thanks for watching. Stay tuned for the next video. Share, like, and subscribe. Thank you.